The Effect of Phototropism on the Growth of Green Beans by Marco, Daniela, Amila, Anna, and Grace.
For the personal engagement portion of this experiment, the overarching question was, to what extent can plants adapt to varying conditions of light, where we sought out to identify that if the plants could grow in different directions favoring a light position, we were curious to know that if they could make that choice, can they learn? So the overall experiment focused on phototropism, which is the movement of plants in response to a light stimulus, and the significance is that it allows for leaves to absorb more light and thus maximize photosynthesis. And so the context for this experiment is that increased photosynthesis results in a slower rise in carbon dioxide levels, which is a greenhouse gas that contributes to climate change. And so by there being an increase in photosynthesis, the plants take in more carbon dioxide, which then results in there being a less amount of carbon dioxide to escape into the atmosphere. And so the hypothesis for this experiment is that if the experimental plants are conditioned to grow towards a perpendicular light, then their angle will be significantly different from that of the control plants grown directly under a light source. And this is because of auxins. Auxins are photoreceptors that are secreted in a plant's stems, buds, and root tips. In order to carry out phototropism, auxins move towards the plant's darker side, the side that is not exposed to the light source, causing the cells there to grow larger than corresponding cells on the lighter side, the side that is exposed to the light source. This then produces a curving of the plant tip towards the light. The experimental plants then will have a different angle compared to that of the control plants, in that by being grown directly under a light source, both sides of the control plants have equal access to light absorption than compared to the experimental plants, where one side of the plant has greater access to light than does the other. The implementation of the results of this experiment can be applied to agriculture, where the species used, more commonly known as the green bean, requires heavy sun and an arid environment to grow in, and do not take much to grow. And so developing regions such as Africa and Southeast Asia feature such conditions. By determining phototropism within these plants and placing these plants in regions that feature such conditions, it allows for an additional food source that's easily grown and accessible, considering that most populations within these regions retain high rates of poverty and are limited to local foods that are often inadequate in nutrients.
In exploration, the following will be covered: experimental design, materials, procedures, experiment, experimental group, controlled group, measurements, and safety considerations. In experimental design, the independent variable is the angle of the light, either perpendicular or directly above the plants. The dependent variable is the angle of plant growth. The constants are the amount of water, half a cup, amount of light per day, 12 hours, amount of soil per plant, 2/3 of a cup, the room temperature and water temperature, which is approximately 22.2 degrees Celsius, and the same species of plants, which are all green beans. The experimental group will be the group with the light perpendicular to the plants. The control group will be the group where the light is placed directly above the plants. The materials for this experiment are 50 mini pots, 25 for each group, measuring cups, protractor, ruler, soil, cold water, 1 box, 2 plant lights, and 50 green bean plants, 25 for each group. The procedures to prepare the plants for the perpendicular light. Place a green bean seed into 25 pots. Take the 25 pots and measure 2/3 of a cup of soil. Make sure to add the same amount of soil per pot. And make sure the green bean is covered. Take one of the boxes and place the pots with the seeds and soil inside of the box. Cut a hole on the right side of the box where the light will be shining through it. Place the plant light perpendicular to the cut on the box. To prepare the plants for the light directly above, place a green bean seed into the other 25 pots. Take the 25 pots and measure another 2/3 of a cup of soil and add that same amount of soil in each pot. Take the plants with soil and place them over a towel. Place the plant light directly above the plants. After following the procedures, the experiment should be something like this. A total of 25 plants are going to be planted into 2 separate groups. Each group is going to be watered approximately half a cup of water each day. Each plant has 2/3 a cup of soil, and the temperature for the water and the room in which the plants will stay in is approximately 22.2 degrees Celsius. All the 25 plants should have access to 12 hours of light each day. To allow the plant to effectively sprout, leave the light on for 12 hours a day and water approximately half a cup.
In analysis, Table 1 depicts the average plant angle per day after 1 week of allowing the bean plants to grow for the experimental control group. As aforementioned, each group had 25 plants. The mean of the 25 plants' angles was calculated per day and per group. Table 2 depicts the standard deviation of each group's angle measurements per day. This will serve to produce a standard error. Figure 1 was made using Table 1's mean angles per group and Table 2 standard deviation calculations for the error bars. The experimental group overall had more variation in its angles than the control group as seen with its error bars. For the control group, as seen with all 7 days, the error bars overlap, signifying that no day was significantly different than the others. However, with the experimental group, days 1 to 3 are not significantly different from each other since the error bars overlap, but days 4 to 7 are significantly different from days 1 to 2, considering that the error bars of days 4 to 7 do not overlap with those of days 1 to 2. All the error bars of days 3 to 7 overlap, signifying that no day is significantly different amongst those days. Day 3 overlaps with days 4 to 7 and days 1 to 2, signifying that it is not significantly different from any day. The chi-square test for this data was performed. The sample size per group is 25, thereby the degrees of freedom is 24. The probability value is 5% and the critical value is approximately 36.415. For this chi-square test, the expected value is the average angle of the control group over 7 days after its 1 week of growth. The observed value is the average angle of the 25 plants in the experimental group per day. The null hypothesis states that there is no significant difference between the control group and the experimental group. Figure 2 depicts the formula to depict chi-square. Figure 3 depicts the calculations for chi-square. The expected value was 88 and the observed value varied by day. The chi-square value was found to be approximately 194.314. This was larger than the critical value of approximately 36.415, showing that the null hypothesis was rejected.
In the experimental group, the light was placed perpendicular to an opening cut into the right side of the box in which the plants reside, and the light was left on for 12 hours each day. To prevent any other light getting into the box and potentially changing the data, we had a lid on the box. This restricted any light from entering the box. The only time the plants could receive additional light was when the box was opened to water the plants. The reason behind why the light was placed at a 90-degree angle was to ultimately test whether or not the plant could condition itself under its circumstances and grow towards the typical light stimulus, and to measure the angle of growth of the plant stem in response to a light stimulus at an angle. The plants in the experimental group were watered half a cup of water each day at approximately 8 a.m. In the control group, the light was placed directly above the plants for about 12 hours a day and received half a cup of water each day as well. The reason behind why the light was placed directly above was to see the angle of growth of the plants towards the light stimulus directly above. The plants in the control group were watered at approximately 8 a.m. each day. When it came to measuring the plants were first measured 1 week after initial growth, and then measured daily for the next 7 days. For each day, the angle of growth of each plant was measured. This was done by measuring the plant stem angle using a protractor and a ruler, and always starting from the soil. Later, the data was recorded onto the table. For safety considerations during this experiment, there was nothing we needed to consider for safety reasons because the growth or the process of growing plants does not show potential threat, especially since the plants were grown indoors. The only danger that could have concerned the group was leaving the plant light on longer than normal, causing the light to overheat, which can start a fire if left unattended. The lack of safety precautions allowed us to potentially speed the growth of the plants and improve the experiment in a few days. It also influenced how much time we left the light on to allow the plants and effectively and quickly sprout. The purpose of this investigation was to conduct an experiment on plants using phototropism to answer the overarching question: can plants learn? The hypothesis inferred that if the experimental plants are conditioned to grow towards a perpendicular light, then their angle will be significantly different from that of the control plants grown directly under a light source. The data ultimately did not support the hypothesis because it shows that there is no significant difference between the control group and the experimental group. The chi-square test was used on the experimental and control group, and the chi-square value was found to be approximately 194.314, which is larger than the critical value of 36.415, showing that the null hypothesis is rejected. Some mistakes that occurred in our methodology include the protractor possibly not being a precise enough tool due to human error in exact measurement. Improvements in our methodology could be having a greater sample size, as it would allow more precise analysis to better depict the impact of phototropism on a plant angle. Computerized angle measurements would also allow more precise measurement of the plant angle, and when measuring in a future experiment, using a precise angle measurement device is something to rely on.
The hormones responsible for phototropism are auxins, which elongate plant cells in the stem, hence allowing a plant to grow towards a light source. Auxins are secreted in a plant stem, buds, or at the root tips. In order to accomplish phototropism, auxins move towards the plant's darker side, causing the cells there to grow larger than corresponding cells on the lighter side. This then produces a curving of the plant tip towards the light. This experiment also connects to the podcast Smarty Pants in that both explore the possibility of plant intelligence. Smarty Pants focused on an experiment where plants were being conditioned, suggesting that they could learn through being dropped multiple times and not opening up their leaves to protect themselves upon learning that there was no need to if it never got harmed. This experiment had a similar approach, where upon being conditioned under a perpendicular light source, the experimental plants were investigated to discover learning, compared to the control plants grown under a direct light source. This experiment also connects to the podcast From Tree to Shiny Tree, in that the root of both the plant and the tree is their main network connection to get nutrients. This allows for photosynthesis in both plants and trees, which have fungi that help them store food. Example is that the trees then supply the fungi with the sugar from the which transfer carbon.